the introduction and also the opportunity to present our research here today. And the topic of my talk today is unlocking low input immunopeptidomics using mild acid elution in a tip. And it was almost correct, Nikolai. We pronounce it MAETi, so mild acid elution in a tip. And I work in the group of Stefan Tänzer, and I'm also supervised by Dr. David Gomez-Cepeda. And I'm really lucky to have them as supervisors guiding me throughout my PhD project. And of course, also thanks to all the rest of the team. Just as a little reminder, so the preprint that we published on this method is already out since last year. Stay tuned, and also after this webinar, we will try to update the preprint as soon as possible with latest data. Okay, but without further ado, I'd like to just quickly dive into immunopeptidomics and immunotherapy. And what you can see here on the right-hand side is actually a T cell eradicating a tumor cell. And the way this works is this T cell senses peptide antigens that are presented on the surface of cells, and these complexes that present these peptide antigens are called major histocompatibility complexes, short MHC, or for the human system, it's HLA. Okay, now for this talk, it's especially important that we take a look at HLA class 1 ligands because they are mainly associated with oncology. They present endogenous protein turnover and, for example, also present viral antigens, or as I said, in the context of oncology. So when you have a look at the structure of HLA-1 complexes or MHC-1 complexes, you will see that they consist, actually, it's a heterodimer consisting of a heavy chain and beta-2 microglobulin, which is non-covalently associated to the heavy chain. This will be important for this method that I'm going to present, so please bear this in mind. And on top of this heavy chain, you can find a peptide binding cleft that typically presents peptides that are in the range of the length 8 to 13 amino acids. And with my intro and the analysis of this peptide repertoire is termed immunopeptidomics. And with my intro, you can maybe already imagine that identifying neoantigens or peptides that are specific for tumors in oncology are really relevant for personalized immunotherapy. So just to get you on track with the methods that you can use to enrich for MHC peptides, traditionally there was a method which was termed mild acid elution. And as the name suggests, you use a mild acid to strip off HLA peptides from the surface of the cell. The acid leads to the disruption of this heavy chain and beta-2 microglobulin, thereby releasing the peptides that are bound to the peptide binding cleft. Afterwards, there are some follow-up processing steps before you go into mass spec. Second, more commonly established method is termed immunoprecipitation, where you usually start from lysed cells and then you capture them via antibodies captured coupled to beads. And then you also elute them from the beads, and then you have the also purification steps afterwards. Okay, so I just like to put this into perspective where we are currently in the field, and especially, for example, also maybe looking into the applications that, for example, with FACS sorting cells or magnetically sorting cells, where we would usually come into the range of required sample input material. And at least for the JY cell line that we are using, and I also think for most other cell lines that you see in the field, for the bulk purifications, mainly people are starting with 1 10⁹ cells as an input, or going down nowadays also. I mean, immunopeptidomics is a technology driven field, and now even going lower and lower. And of course, as I said, immunopeptidomics is technology driven, and there has also been some nice work done on integrating microfluidics in the immunopeptidomics sample prep, thereby even approaching lower required input materials. Okay, what I'm going to present you today in this webinar is a method that we came up with. As I said, it's termed MAETi, mild acid elution in a tip. And with this method, you can actually, even without microfluidics or a complex setup limiting throughput, you can even acquire cell inputs that are even lower to currently reported microfluidics input numbers, which would potentially make it compatible with FACS sorted cells or sorted cell populations. And I think it's really exciting to be in immunopeptidomics at the moment, because if we think of a heterogeneous tumor microenvironment, there are also different cells involved. And I think it gets really exciting when we come to cell subpopulation immunopeptidomics. Okay, so just to get you on board with the method that we developed, in the usual bulk mild acid elution protocol that we adapted from Sturm et al., it consists of many steps. One part of this protocol is that sequential centrifugations are used to remove potential debris from the cells that you would have. And also this is set to increase the proportion of these short 8 to 13 minutes that are potentially to be presented by HLA complexes. You can see there are many steps involved, including desalting, lyophilization, and also resuspending prior MS analysis. And now the method that we came up with is that we directly load cells on a membrane-tapered low-bind pipette tip, and coupled with the Evotip technology, we can circumvent a lot of steps that are required in the bulk mild acid elution, and thereby boost sensitivity of our method. Okay, so here the method is outlined again, and in the first proof of principle, we stick to a citric acid-based mild acid elution buffer that is also featured in the Sturm et al. Paper from 2021, where they compare immunoprecipitation with mild acid elution. And what you can see here is, as I said, we loaded the cells on a pipette tip that is tapered with a glass fiber membrane, and then we strip the cells directly into this Evotip and separate it on our timsTOF ecosystem. Okay, and in the timsTOF, you can typically design polygons for precursor selection, for fragmentation and identification. And for example, you would maybe call this polygon a step polygon. And here protected are all short peptides with the length 8 to 13 amino acids. And you might have heard that our group has also some history in HLA-tailored immunopeptidomics methods. And what you can see here is that we tailored the polygon specifically to our needs for HLA peptides. But we wanted, in the first proof of principle with the citric acid-based buffer, we wanted to compare the two methods and also enabling unbiased evaluation of our protocol. Okay, remember I said those peptides are typically in the range 8 to 13 amino acids. So for us, it's also interesting to take a look at the length distribution of peptides that we enrich with our methodology. And what we can see here, irrespective of the method that we used, we had a nice length distribution with the apex of 9 amino acids that is usually to be expected for HLA class 1 ligands. Also, the proportion of 8 to 13 mers accordingly was really high, with slightly higher 8 to 13 mers for Thunder compared to the step polygon. Overall, this yielded, on average per 50,000 cells, almost 1,000 8 to 13 mers using Thunder DDA-PASEF. And also, when we take a look at the data completeness, where we have a look at how many peptides are identified across all replicates, we can also see a slight improvement for Thunder DDA-PASEF, but not very significant. Also, when we have a look at the MS1 area coefficient of variations of the short 8 to 13 mers, we can also see that the reproducibility of Thunder DDA-PASEF was slightly better, but irrespective of the method, it was around 35% mean area CV. Okay, and in the setup, among the 8 to 13 mers, we identified over 75% of predicted binders that actually also followed the motifs that we would expect for our tested cell line JY here. Okay, so next we approached this whole methodology moving forward with higher sensitivity instruments, and also among the instruments that we use was timsTOF SCP. And we also noticed at the start while testing citric acid that alone the citric acid buffer substance alone showed high traces of contaminant ions, which is definitely not ideal for a high sensitivity protocol. And so we started our journey to investigate alternative buffer substances that are buffering at a similar pH of citric acid. And among the substances that we tested, beta-alanine was performing really well. And also, if you see for the pure buffer substances here shown in the heat maps of ion mobility versus retention time, beta-alanine was considerably cleaner compared to citric acid. Okay, now having a buffer substance that is considerably cleaner, we moved on with the actual MAETi protocol and compared citric acid with beta-alanine. And what we found here was actually quite remarkable. Irrespective of the buffer substance that we used, we got a nice length distribution of 8 to 13 mers, where beta-alanine was not performing worse than citric acid, for example. But the striking thing was actually that if we come to 8 to 13 mer identification on average per 50,000 JY cells, we actually have a boost of almost 50% or over 50% of 8 to 13 mers on average identified per 50,000 cells. The data completeness was also a bit better for the beta-alanine protocol, and also since we have less noise in the background of the buffer, the quantitative performance here in MS1 area CV of 8 to 13 mers was improving with this improved protocol that we had. Okay, for the predicted binder, we also identified significantly more binders for beta-alanine with overall quite a high overlap here. This data was acquired in DDA, and most importantly, we have the same motifs retrieved by using either citric acid or beta-alanine. So next, having established or tested this beta-alanine substance as an alternative to citric acid on a small scale, we wanted to really make sure that, for example, comparing it to immunoprecipitation, that we are not introducing a bias in our protocol. So we set out in the lab and started with JY and Raji cell lines. These two cell lines are B cell lines, but they share no alleles. So no peptides that are supposed to be presented by the alleles are supposed to be shared between the two cell lines. We subjected it to the bulk protocol starting from 40 million cells per pellet and injecting it 3 times with this step DDA-PASEF method, again, to also select potential precursors that are outside the range of 8 to 13 mers for unbiased comparison. What we found here is that irrespective of mild acid elution or immunoprecipitation for both JY and Raji, we had a really nice enrichment of 8 to 13 mers. And if we look at the average identification of 8 to 13 mers between IP and mild acid elution, for most cases, mild acid elution was performing almost the same as immunoprecipitation, but for Raji, even though the overall proportion was slightly lower, we identified more 8 to 13 mers, even with the mild acid elution protocol using beta-alanine. Okay, for the predicted binders, it's actually more or less the same. We can see also here for Raji mild acid elution, it was slightly lower, but overall for the average identification on predicted peptides or predicted binders, mild acid elution was still performing slightly better for JY and Raji. Both yielded approximately the same numbers of predicted binders. What is really important is that actually the overlap between immunoprecipitation and mild acid elution was really, really high. Again, this is DDA data, so there might be some variability, but over 70% overlap is really high. And needless to say, they also followed the same peptide binding motifs. So this, for us, showed that we actually can use this beta-alanine mild acid elution without introducing a bias, and also establishing the rationale that we can use it for this low input scenario. And just to get you also on board with what protocol formats we can offer with this MAETi method, we can either, for highest sensitivity, directly load the cells on a tip, but as you can imagine, this tip might clog if you just load 1 million cells on a tip or so. So that's why if you go beyond 100,000 cells, we would recommend that you do your protocol in a tip, strip them, but later on still transfer them to this glass fiber tip and directly into the Evotip to retain some remaining debris. And what we did here is actually we did a profiling titration of JY cells, going from 25,000 cells to 1 million cells, just to showcase also these two protocols that you can use, and also introducing versatility in your way, how you might apply this method. Okay, so next, in order to establish some more biological context, we teamed up with Dr. Claire Conch from the immunomonitoring platform in HI-TRON Mainz, and we used PBMCs. It's a readily available source of biomaterial. We purified PBMCs and we subjected it to magnetic activated cell sorting, enriching for the B cells. We also had the non-B cells still sticking on the magnet, so to say. So I termed this population here B-negative population. I subjected them to the plate protocol here, 250,000 and 500,000 cells per well, and we did the MAETi protocol. And interestingly, now moving away from cell culture, so to say, into biological application, we again had a real nice length distribution of the cell population that we targeted, having a real nice 8 to 30 mole proportion here in the length distributions. The B-negative cell population were not so specific, but still having an apex at 9 mers. And yeah, so overall for the targeted cell population, the enrichment was really specific. So now if we take a look at the predicted binders that we recovered here in these methods, we can actually see that again, we did some analysis in HLA-tailored DIA-PASEF and also Thunder DDA-PASEF, and DIA again, or of course, was performing better than the DDA workflow. And for example, if we now take a look at two populations where we identified almost the same amount of peptides on average, and check for the overlap, we can actually see that here in this DIA approach, they shared approximately 50% of predicted binders between the B cell and the B negative cell population. And of course, you can also be interested in now comparing it against this background of B negative cells. And that's best if you can do it now here with the differential analysis. And indeed, for example, if we look at the log 2-fold change of B cells against B negative cell population, you will find some interesting hits here. For example, representations here of the class 2 antigen presentation networks, which is also reflected in the string analysis here. You can also see prominent differential ID popping up, which is P53, but there are also B cell specific or associated factors such as PTN6 and BANG6. And overall, this describes the cell type that is actively preparing or engaging in immune responses, and particularly emphasizing on antigen presentation and B cell activity. And indeed, that's also what we get if we take a look at the gene ontology processes of the strongest pathways, where we have antigen presentation, but also some lymphocyte associated pathways. So which would really nicely reflect an antigen presenting cell such as a B cell. So yes, coming to the conclusion of my talk here, first we showed a proof of principle that this mild acid elution in the tip is actually possible, starting from as low as 50,000 cells. In our case, we were using JY cells, and with the small tweaks of testing alternative buffer substances, we found beta alanine that was actually performing better than citric acid-based buffers. And also importantly for the robustness for this method, we compared bulk acid elution with immunoprecipitation, and both methods identified complementary immunopeptidome profiles. And last but not least, we also showed that MAETi can be applied, for example, to sorted cell populations. In our case, we showed this magnetically activated cell sorting. And the last steps of the method optimization are still ongoing. We're cooking up something nicely for the manuscript. And so, yeah, just stay tuned what we will be coming up. And applications of these methods for sure can also include drug screenings or also doing kinetics where you would be previously using a lot of cells and need to grow them. Yeah, and our dream is still also advanced in oncology, advancing, for example, tumor microenvironments and analyzing cell populations here. Okay, with this, I'm at the end of my presentation, and I thank all the team of Agitense, also David, and also Evosep for providing me with the opportunity to present our work here. And with this, I'm at the end and would give the screen to Bob then.